Hello, everyone. Welcome to today's live panel discussion, Promoting Reproducible Animal Research in Journal Publications. I am Brenda Kelly Kim of LabRoots, and I'll be moderating today's discussion. We're delighted to bring you this panel discussion presented by LabRoots, the leading social networking site for science professionals. For more information, please visit us at labroots.com. We have a few important announcements before we begin. This discussion is designed to be interactive, and we want to hear from you. Ask questions. You can submit your questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or alternately, submit your problem through the green Q&A button on the lower left. I would now like to introduce today's speakers, Susan Compton and Linda Toth. Susan Compton received her PhD in microbiology from the Uniformed Service University of the Health Science and completed a postdoctoral fellowship at the University of Colorado. Dr. Compton joined the faculty of the section of comparative medicine in the Yale University School of Medicine in 1991 and has been their chief of the molecular, serological, and virological diagnostics unit since 1998. Susan has also held several editorial positions within the American Association for Laboratory Animal Science and is currently the associate editor of the Journal of the American Association of Laboratory Animal Science. Her research is focused on the diagnosis, pathogenesis, transmission, and prevention of viral infections of rodents, in particular coronaviruses, parvoviruses, noroviruses, and most recently, astroviruses. Dr. Toth received her PhD in pharmacology from the University of Pittsburgh in 1980 and her DVM degree from Purdue University in 1986. Since 2004, she has held the position of Associate Dean of Research and Faculty Affairs at the Southern Illinois University School of Medicine, where she is also a professor in the Department of Pharmacology. From 2000 to 2006, she was director of the Division of Laboratory Animal Medicine at SIUSM. She is also an active member of AALAS, the American Association of Laboratory Animal Science, and serves as the editor-in-chief for that organization's two peer-reviewed journals, Comparative Medicine and the Journal of the American Association of Laboratory Animal Science. Finally, she is an appointed member of an NIH study section, served on an NIEHS advisory committee, is a steering committee member and past chair of the Group on Research Advancement and Development of the Association of American Medical Colleges, and is an advisor to the Executive Leadership and Academic Medicine program of Drexel University. Dr. Toth's research interests focus on the genetic modulation, sleep propensity in healthy and disease, the impact of sleep loss and diurnal disruption on susceptibility to and recuperation from infectious and inflammatory disease, and the impact of environment on research animals and data. I will now turn it over to Drs. Compton and Toth to begin their discussion.
Thank you all for attending. I see we have a reasonably large number of attendees, and Sue and I are going to attempt to give you our perspective on promoting reproducible animal research in journal publications. We bring this presentation to you with the background of over 10 years of experience as journal editors for ALAS and its two journals. We also have been reviewers on many other journal articles over the course of the years, and we are also functional scientists, so we understand this issue not only from the perspective of journal editors, but also from the perspective of those who are trying to replicate both our own research and that others have published in the literature. Sue, would you like to say a few words?
No, that pretty much says what we need to know at this point.
Okay, so we're going to present a little bit of background here based on our perspectives. And after the quick presentation, then we will take questions. So all questions are welcome. Please let us know how we can answer your questions with regard to reproducible animal research and perhaps publication in general in the laboratory animal field. So I would first make the statement that as journal editors and as scientists, what we want to do is fundamentally help communicate our science and make it better. And journals offer a forum in which both of those activities can take place. Not only do we try to publish sound science, but we also try to act as editors as a feedback opportunity for authors. My personal view is that in many cases, reviewers are viewed as negative rather than constructive. And although that may seem to be the case, fundamentally, it's the author's responsibility to convince the reviewers that their work is sound and publishable. And any feedback that comes from reviewers should be taken in the sense, in my mind, of offering a constructive critic, offering a problem that someone has perceived with your work. And the point is to get that problem fixed before it goes out to communicate, before it goes out to be communicated to the real world. So we have two learning objectives in this session. First of all, we're going to overview what factors diminish the reproducibility of published research. And at the end, we'll talk about how journals can promote reproducibility through their publication requirements. So first, to define reproducibility, I've gone to the standard source of Wikipedia for their definition, and basically that is the ability of an experiment or study to be replicated, either by the same researcher or by someone else working independently. So reproducible research refers to the idea that the publication is the final product of the research. That is what is disseminated to the public. And it should provide all information that's necessary to reproduce the findings and build on them. And that can include not only animal-related details, but also bench science information, the reagents that you used. For example, if you're using database work and doing database work and you had to write code to analyze the data, the code should be available. Basically anything that anyone would need, to your knowledge, to reproduce what is being done. Now, the first stage of reproducibility is within the PI's lab, and it's fundamentally important that whoever is doing a study should be absolutely clear that they can reproduce their main finding. This often is not, proof of this is generally not demanded by reviewers. It's generally assumed that if you are submitting something, it is reproducible in your hands, and that the effect is strong, reliable, and durable. So we really, I think, go on the assumption that if something is submitted for publication, that the author is very confident that they can replicate it in their own hands. I'm not sure how often, though, many individuals really follow up on this. One-time observation with a certain N may be deemed appropriate without necessarily repeating an entire study to make sure that what you've got can be recreated in your hands. So I think what we're really talking about in terms of publication is that we assume that the information is replicable by the author, should also be replicable by others. Now, this is not always the case. We have had at least one instance in the ALAS journals where a group of authors communicated to us that they could not reproduce their original finding. And I really have to applaud them for that, because they realized and acknowledged that even though they thought they had a sound finding, that it was not as substantial as they expected it to be. And in any case, notifying the field that there's an issue that the authors don't understand and cannot solve is an important step to preventing resources from being moved into a wrong direction. So I think that that kind of disclosure is critical to being sure that we don't waste resources that are available to us. So some of these I'm sure you've all heard of and thought of before, and we'll go through them kind of quickly because I think that these are factors that we're all well aware of. I will say, first of all, that in terms of reproducibility of science, it's not just animal research that's the problem. Many kinds of research, many research studies, whether they're strictly in vitro studies or extend into the human realm, are difficult to replicate. And that happens because when we're doing research, we don't necessarily understand everything that can impact the system. And we'll give an example of this later, but essentially we study a system based on the knowledge that we have at hand, and if there are either factors that we can't control or factors that we don't even know about, these may influence research differentially as different groups try to repeat the same basic study. So in many cases, it's hard to put a number on it, but inability to replicate does not necessarily imply that someone has done something wrong. It can also be that there's something there that we don't know about that's influencing the system under interrogation. So this is an important thing to think about when you try to replicate someone's study and can't. We don't know everything about the system that we may need to. All right, so moving on from that, here are our experimental design factors. You've all heard of these. Incorrect group sizes, and I might say, usually in my mind, inadequate group sizes. You see many studies in the animal literature that are trying to conserve animals, sometimes to a level that seems inconsistent with answering the question that is being asked. And assuring that your group size is appropriate is really critical to being sure that you have sound, replicable data. This may mean repeating an entire study, or it may mean powering your study initially to be sure that you can not miss or not miss real effects, or that you do not overinterpret a small one-time effect. Control should be appropriate. Basically, you want to control for everything that you can except for the one factor that you are manipulating. You want your groups to be identical. And this is perhaps easier said than done for some reasons we'll talk about later, but maintaining your animals in strict parallel circumstances throughout the course of the study is really important. Blinding and randomization to groups are also common problems. Blinding is important, particularly, I would say, with regard to subjective assessments of outcomes. And so if you are using a study in which you're somehow evaluating your animals for pain or some other type of subjective assessment that having the observers be blinded to the condition of the animal is really important. Randomization is something that I think is less emphasized in animal studies, but maybe should be. We have a tendency when we're doing experiments to assign animals to groups based on which ones we pick up in the cage. And doing that may create an unintentional bias if some of the members of a group are more active, more mobile, harder to catch, easier to catch. So if you use a systemized randomization scheme, that can be avoided. And also, for all statistical and experimental design questions, you should plan in advance what you're going to ask of the data and how you are going to analyze the data. And that is really fundamental. You can't fish for significant, statistically speaking, you shouldn't go fishing for significant results unless they are part of your experimental design with pre-planned questions and pre-planned analyses. Other factors that diminish the reproducibility of animal research we're probably well aware of: lack of housing and husbandry details, the information that's necessary to identify the source of the animals and their various characteristics, and where, when, and who the procedures were performed. And this type of information is, I would say, particularly salient because there have been a number of studies out there where individuals at groups in various laboratories have absolutely tried to standardize everything they could with regard to how the study was done, and then perform studies that were attempting to replicate traits that they found in various strains of mice, and were able to replicate some of the effects, but not all of the effects. So clearly, even in very experienced laboratories who understand and appreciate all of these sources of variation, it's still hard to determine what the factors are that are really influencing the behavior or perhaps the physiology of the animals you're studying. And a good example of this was revealed perhaps last year or the year before by the Mogul Lab, which does pain research, and they presented the very amazing, really, and well-documented report stating that the sex of the technician who performed the test procedures had an outcome on the pain response of the animals. That is, if males perform the procedures, male researchers perform the procedures, the test procedures, the mice showed stress-related analgesia that was not present if females performed the procedures. So there's a case of something that probably none of us had ever thought of as a factor in some of the testing, but in fact when this very observant and highly experienced group of scientists did this study, they were able to notice that bias in their data. So I think that just illustrates how careful you can be and how you can overlook things that may be important in replicability, not through necessarily lack of attention to detail, but simply by not knowing what kinds of things the animals that we're working with will respond to. Okay, sample size and group size is a really important issue that I feel we have undervalued in animal research. In human research, power analysis and a predetermination of group sizes is just the expectation, the basic expectation of what is going to happen. But in animal work, we typically at least in many places, seem to overlook that, including by presenting the power analysis in publication. And that's something that I think we can rectify or demand either as animal care and use committees that are approving these studies, or as on the publication side as requiring a power analysis to justify group sizes. Because this is really critical. You see some very complicated studies out there that have very low numbers of animals per group. And with those few animals and the many variables that are being measured, it seems pretty clear that sample sizes are inadequate. Another important thing to consider is whether the experimental unit is the whole cage or an individual animal within the cage. So if you have a group of 5 and they're all in the same cage, the problem is that if something happens to that cage, it can influence that cage and bias the outcomes based on which group that cage of animals belongs to. So, for example, if you have one cage that's your experimental group and that cage happens to experience a water leak, or there's a lot of fighting in that cage, or maybe there's some clinical illness in that cage, that can bias the outcome toward animals in that cage and give you an outcome that is dependent on some factor other than what you're trying to test. And if all your animals are in the same cage for one group, that's really a problem. And then your number, your N, might be not 5 animals, but one cage, or however many cages you've assigned to that condition. So sometimes it's hard to work with that within the confines of space and cost, but it is a really important avenue for biasing the data. And I will also say that I think this also applies to bench work. In many cases, when you do bench work and you run a triplicate, if you run your triplicate in series, you're going to have a lot less variability than if you run them randomly throughout your plate. And this kind of variability is something that's, or potential for variability is something that's not generally, in my experience, not generally recognized or acknowledged. So running your triplicates in series is definitely going to make your, well, in my hands anyway, it's going to make your values tighter than they might otherwise be. Okay, we've really talked about this in some way, about controls and randomization. Certainly for randomization, you can use random number series to assign group numbers to treatment groups. For subjective data and blinding, you have to ask the questions of not only will the person interpreting the data or generating the data be blinded to the study groups, but is one rater enough? And if you have more than one raters, how are you establishing uniformity across raters? And there are approaches to doing this that should be followed. This applies not only to animal research, but also to any type of qualitative research. When you're evaluating subjectively in a qualitative manner, having multiple raters and documenting uniformity across raters should be established. And you can see this done. Long ago, I remember one example where we had a group of surgeons and the surgeons and the surgery residents who were evaluating endoscopy photographs, and they established a scale for how they were going to evaluate the severity of the lesions that they were observing, and tested themselves. And even the individual who generated the scale got a poor score, so was not even replicating himself. So being sure that you have established a scoring system where your raters are equivalent or can be tested is really important for making sure that your data, the data you collect, are valid. For housing and husbandry, factors that are fundamental to replicable research are the frequency of housing, whether the animals are group housed or singly housed, what types of enrichment are provided, of course the food, water, bedding, and cage type, and even where the mice were housed in the room or on a rack can have an influence, partly because environmental conditions can change. Lighting conditions can be different. Ventilation might be different. A lot of factors of that type influence how the animals assigned to a study can react to either the experimental or the control condition. The fourth bullet there, cage type, I think we've been recently really received a lot of good data on that point from the studies of Bob Douchy and Melissa Wren, who have shown that cage color or tint significantly influences some physiologic parameters in the mice housed in the cage. And if you are from a facility that uses colored cages, or that, even worse, uses colored cages at random for different groups of animals, you may be introducing variability and a sense of irreproducibility into the study. Enrichment, I believe, is something that should be very carefully monitored and standardized and reported, so that the environment of the animal for the duration of a study should be controlled. One of the big advantages of using animals in research is that we can control their environment. And enriching their environment is fine, but it should be done in a standardized, reproducible way that is reported in the publication, because there are many studies out there that show that enrichment definitely can influence the physiological or behavioral responses of the animals. And if your readers don't know what you have done, or even worse, if you don't know what you have done, it may be very difficult to reproduce that type of an environment. In terms of single versus group housing, you can run into the issue of beginning with group housing, and then as the study proceeds, animals drop out. That clearly changes the environment within the cage for the animals that are remaining, and so that is another issue that should be considered when you're designing a study. The description of the animals, you've mentioned a number of things here that I think probably many listeners are familiar with. The strain designation is the one that I will mostly emphasize, because our genetic backgrounds are getting more and more complicated, and the appropriate nomenclature is just essential. I personally had a circumstance where I had submitted a paper for publication, and I uniformly referred to the animals that I used as BALB-C BYJ. And one of the reviewers of my paper asked why I didn't use that designation throughout the discussion, and that was because many of the papers that I read just said BALB-C, and so I could not extrapolate if they were using the same source of animals as I was or not. And as many of us know, that can have an impact on the outcomes. Strains of animals, even inbred strains that you get from different vendors, can clearly have different genetics due to genetic drift, and so it's important to know about that. The genetic manipulations also can interfere with the reproducibility of research if the genetic background influences the research outcomes. And there has been an idea propagated that using a mixed genetic background is a good thing, and in some circumstances it may be, but remember, the point of this research is to control everything you can. The point of animal research is to have as much control as you can on the animal. And if you are going to allow variations in health status, or variations in genetic status, or variations in housing into your studies, that is going to impact your outcomes in ways that you probably can't predict. experimental procedures, time of day that the samples were collected or the behavior was observed. This can be really critical, as there can be large changes due to circadian variations in various measurements or behaviors. And then other experimental procedures that I've mentioned here, source, dose, and route of agents that you administer, how you monitor for effects, and how you define subjective scales. I see that I'm using a lot of time, so I'm going to try to speed up here a little bit. Statistics, I already mentioned, should be pre-planned with pre-planned questions, a power analysis, and a plan for data analysis. And in my mind, it is always important to include a statistician on the research team, both as part of the design phase and as part of the analysis phase. I also would urge everyone to try to evaluate biological as opposed to simply statistical significance. So if you have a value and it goes from low to lower, maybe it's statistically significant, but is that biologically meaningful? That is worth some consideration. It's not really a point of reproducibility, but it may be a point relevant to the significance of this work that you're studying. And I will add to that what I've heard from, can't remember where I got this quote, but from a statistician: if you torture the data long enough, it will admit to something. So if you design your study properly, you should not have to torture the data. You should find the result that you find, or not find the result that you're looking for in the context of your immediate and your fundamental experimental questions and experimental design. So in terms of reproducibility, this can be aided by clear publication requirements. As I mentioned earlier, as we review manuscripts for the ALAS journals, we assume that they are replicable by the submitting authors, and we rarely require evidence of a validation study, and I think that is generally true. In my mind, the responsibility there lies with mentors, largely, that mentors should have high standards to impart to their trainees in terms of habits and practices that they teach their trainees are adequate for performing valid research. High standards in mentors are really important, and it's hard for me to say anyway, whether that's a problem or not, but I've certainly seen at least some instances where it seems to be a problem. Journals can promote reproducibility by requiring a complete method section, and with regard to animal research, probably I would say that the Arrive guidelines, which maybe you all have heard of, and this guideline supplies a very focused and thorough list of factors that I have reviewed and other factors that should be considered in developing a methods section. Over the past years in methods section development for scientific publication, I would say there's been a tendency to diminish the importance of the methods section and put it at the end, or put it in an appendix, or put it somewhere where it's not obvious. But to me, that is really a strange attitude, because how can you really understand the paper if you don't understand their methods? That's just fundamental to collecting the data and knowing how to interpret the data. So it is not a cut and paste type of activity in my mind. It's something that really needs to be thoroughly vetted from all perspectives. If you didn't know the method, could you repeat it based on what's in that article? And at the ALAS journals, we have reference to the Arrive guidelines for authors and for reviewers, so we hope that those are helpful. And so finally, let me just point out that the real peer review starts after publication. We attempt to provide peer review as part of our review process for the journals and their articles, but we are not the end of the process. The publication is where the peer review starts, when people try to replicate your work and can or can't, or look at your work and find flaws that then reflect on you and on your science. So I would just urge you to remember that the peer review process for publication helps you to avoid problems with peer review that will occur after your work is released. And this is a very critical service that I think should not be viewed negatively in most cases. So with that, I will stop and see if Sue has anything she wants to add, and then we'll take questions.
What I would like to say is you really can never put too much in the methods. So if you think that there's something that's important for somebody to reproduce it, put it in there. No, somebody doesn't have to give you a list of what needs to be in there. Everything you think that was a variable. And don't be afraid to say something went wrong during the experiment, that there was an unexpected outcome because you had, a series of fire alarms, or you had a series of water bottle accidents, or there was an outbreak down the hall and you had to treat with ivermectin or something else. Put all of that in the methods, because you don't know when you write it if there's going to be a variable, or if somebody else is going to recognize that as a variable that they've seen before and say, wow, they saw this and I saw something similar when we had the same problem. All of those things are helpful when somebody's trying to interpret your study.
Great. Thank you both. Thank you both. Is there anything else you want to continue to talk about with the audience, or should we have some questions?
I think we're ready for questions.
Awesome. Yes. Let me just remind our audience after this presentation on how they can submit their questions. You can submit questions by typing them in the green Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We're going to try to get to as many of your questions as possible. But again, I just want to thank both Dr. Toth and Dr. Compton for such a great presentation. It really does speak to how much is involved in creating your studies so that they are worthwhile to your community. I'm going to give our audience just a few seconds to bring up the Q&A box, typing in any questions that they might have, and we'll get on with that. And don't be shy, because Drs. Toth and Compton definitely want to hear from you.
And I would say if you have any questions related to publication in the ALAS journals, we'd be happy to take those as well.
That's great. I'm sure with this group here, there's probably a lot of studies going on, and knowing exactly how to have those come up for review is important for our audience.
Anybody out there? There must be someone who has a question or something we can help you with. Or at least give our...
Let me see here. We do have one question coming in. Do you find that there are problems with animal use common in submitted manuscripts? Is this a common occurrence for you?
Sue, would you like to comment?
What was the question again? Was it?
Are there problems with animal use common in submitted manuscripts?
We don't see an awful lot of problems in the ALAS journals because most people are very knowledgeable about working with animals. I'm more likely to see it in more categorical science journals where there's not a lot of information on the animal methods, and it's more frequent to see, people abbreviating strain names and other things of that nature, and not understanding that animals are a complex organism, and it's not the same as working with a cell line. And it's the people who are transitioning that sometimes don't understand and put enough methodological details into their paper. But most people who work with animals every day don't seem to have that problem.
Great.
We do have. Our reviewers are very good at catching those omissions as well. So, basically, I think that in the ALS journals, we tend to cover a lot of that information. There was a study, someone did a study last year that they gave a poster for at ALS where they did evaluate 2 or 3 years of publications in JLAS about animal use and how we complied with the ARRIVE guidelines. And I don't remember the exact data. I'm hoping they will publish that as a full manuscript. But basically, I think we did okay. Not everything. We didn't capture everything that was in the ARRIVE guidelines, but had a very, very reasonable report card on that. Is that your remembrance of that, Sue, as well?
Yes.
Great. We have another question from an attendee. As editors, how do you look at studies that have negative results? Often these are not published.
Yes, I would say that we publish many negative results because many of the studies that we publish, particularly in JLAS, deal with testing various regulatory guidelines and demonstrating whether they produce an impact on the animal that's different from other conditions. So basically, I would say we publish a lot of studies that find no difference between one situation and another situation. So I don't.
Great.
Would you agree, Sue?
I agree. I think because we're trying to get basic information on how to best use animals, most studies, whether they're positive or negative, will add to information that can be used by other veterinarians, by IA cooks, and other things of that nature. So I think we're more willing than some of the other journals to publish either way.
Yeah, I think the most important thing there is not whether the results are negative or positive, but whether it's an important question. So if it's a research question that many people are, or an animal use question generally, that people are struggling with, having data to show that something does not make a difference can be very beneficial.
Great. We do have another question. Are there any specific recommendations related to research with zebrafish?
Specific. I am not personally, I don't know. I think that the general the general requirements for publication in a methods section are very similar. Certainly fish are going to have different environmental requirements than other types of animals, but as far as describing thoroughly the environment and the experimental approach, I think that is, in general, very similar.
We don't publish a huge amount of publications on zebrafish, and in general, we would ask for the same kind of information so that we would know all of the husbandry and housing and handling kind of information that was used in that study.
Probably the environmental controls, the quality assurance factors for zebrafish are maybe an area that has not been as well developed as it has been for rodents. So that might be an area where health assessment and quality assurance could use some work to give some of you some ideas for perhaps publishing or doing research and then publishing information. I don't think there are a lot of strong guidelines out there, at least not in the published literature for that I'm familiar with as to how to provide that kind of quality assurance.
Great. Moving on to our next question. One of our attendees asks, I apologize that I only heard the last half of the presentation, and perhaps this has been addressed, but should the info presented today compel our scientific societies to establish detailed SOPs for conducting some of the more common studies in their fields to help reduce variability across different investigators and institutions, et cetera?
It's almost a philosophic question. You know, I believe that certainly there may be preferred ways to doing things, but I personally am not too inclined to stifle scientific ingenuity by putting too many controls on how people design their experiments and what they do. If there can be some standardization, then that doesn't, there's no really wrong side to that, but as far as making it requirements, I would just have to think very carefully about something like that. My call would be more to have clear reporting rather than clear requirements. Did that answer your, maybe I didn't quite get the question.
We do have another question, moving on to, what are some effective ways to include the negative results, which you mentioned can be useful for future studies, in a way that does not prevent the acceptance of the manuscript by reviewers?
You have to present the information in a way that shows that the negative result is useful. You know, negative results can be very useful. For example, if you compare two diets and you find no difference, or if you compare two cage sizes and you find no difference in animal well-being, that could be very useful information. So what's important is not that it's negative data, it is that the question is an important one and the answer is useful no matter which way it turns out.
Okay, we have another question. What are the most common reasons for rejection of submitted articles?
Sue, would you like to take one?
I think the most common is having a poor experimental design, so having a sample size that's too small, or possibly extending the conclusions beyond the data. So essentially having data that doesn't really show results and trying to say there is an effect, or saying that the effect is applicable to all animals when all you worked with was a CP7 black male. And so you need to make sure that your conclusions are not overreaching. And also, if you have to make sure that when you're looking and saying something has a significant effect, if it's not statistically significant, it's not really significant. And that probably means that your sample size was too small. And so saying there's a trend towards something means you should probably run a follow-up experiment with a larger sample size to see if the effect is true.
Okay, thank you. We have the next question. What should I do if I cannot reproduce published research?
Well, the first thing that I would suggest, if it's research that you published, you should contact the journal. And we will publish as a letter to the editor that you explain to the research community that you tried to reproduce your own work and you were unable to do it for unforeseen reasons. And to me, that is a good way to maintain integrity in the scientific method. If it's published by someone else, the person to contact is the research group and see whether you are really replicating or if something is missing from the published methods section that you were trying to replicate. That's really the best step, is to just make sure that you have covered all your bases in terms of the fact that what you are doing is exactly equivalent to what was done in the published work. Though I would also keep in mind that, as I mentioned earlier, there are many subtle influences on animal-based research, and we don't always know what their magnitude is or how they can expect the let's say, unknown variables to influence outcomes. So if you have something that you have tried to replicate and you can't, that's not necessarily a non-publishable finding. I think it is a place where we need perhaps more research to find out what the source of that variation is.
Okay, thank you for that. We have another question from an attendee about the amount of information in the animal section. He asked, can all of this information be in one paragraph?
No, I don't think one paragraph would do it, but we don't have any limitation to article sizes in our journals, at least. So that might not be true for every journal, but in the ALS journals, you can put as much detail in there as you would like to.
I mean, it's not uncommon in the ALS journals for the materials and methods to be longer than the results.
That's very true, right?
Okay. I think that is all the questions that I've seen coming into the Q&A. I would like to thank Drs. Toth and Dr. Compton for making this presentation possible and for bringing their information to us today. Doctors, would you have any last final comments for our audience on your topic?
Well, I guess I'd go back to my last slide. The real review starts after you publish, and I would say keep that in mind and try to view your critique of your manuscripts as the first step in preventing misunderstanding between what you published and what you meant to say.
I'd say that planning the experiment is as important as analyzing it afterwards. If it's well planned, the analysis is a lot easier to do. Great. Thank you both so much. I'd also like to thank LabRoots.com for making this panel discussion possible.
Thank you to all the attendees.
Thank you.
And well, to our moderator.
We are happy to be here and bring this information to you. It's going to be available on demand through August of 2016 in case anybody wasn't able to come in and listen live. Attendees will receive an email from LabRoots alerting you when this webcast will be available for replay. And we would invite you to forward that email to any of your colleagues who may have missed today's live discussion. Thank you again for joining us. Thank you, Dr. Toth and Dr. Compton. We hope to see you next time. Thank you.
Thank you.
Thank you.